Science has a graveyard. It's full of things we were absolutely certain were true, until a single discovery walked in and collapsed everything. Not gradually, overnight. The kind of finding that makes a researcher put down their pen, stare at the wall, and whisper, "Everything we thought we knew was wrong." These are not ancient mistakes. Some happened last year. But here is the thing nobody tells you. The most disturbing discovery on this entire list generated over $50 billion, saved hundreds of millions of lives, and was taken from a person who never gave permission and died never knowing any of it happened. And by the time you get there, the science will be the least unsettling part. Join us, and let's discover the 13 scientific findings that broke what we thought we knew. Let's go.
Number 13. Rising Star Cave. In 2013, a recreational caver named Stephen Tucker squeezed through a crack in a South African cliff so narrow that only his exhaled breath could fit through it sideways. What he found on the other side changed the entire story of what it means to be human. Deep inside the Rising Star Cave system, Tucker discovered a chamber containing over 1,500 bone fragments belonging to a previously unknown species of ancient human, now named Homo naledi, the largest collection of hominin fossils ever found in Africa. The creature had hands built for tool use, feet shaped for upright walking, and a brain roughly the size of an orange, about 1/3 the size of yours. That combination should not exist. Science had always assumed that complex behavior required a large brain. Small brain meant simple animal. That was the rule. Homo naledi broke it completely. But the finding that stopped researchers cold was not the anatomy, it was the location. The bones were found in a chamber with no evidence of water flow, no predator activity, no signs of any natural process that could have moved them there. The only way those bodies entered that chamber was through deliberate navigation of an extremely difficult underground passage in complete darkness. Researcher Lee Berger from the University of the Witwatersrand concluded that Homo naledi, a small-brained creature that existed hundreds of thousands of years before modern humans, was intentionally placing its dead in a specific underground location. Intentional burial of the dead was supposed to be exclusively human behavior, tied directly to large brain development and abstract thought. A creature with a brain the size of an orange was doing it long before we existed. Every textbook assumption connecting brain size to complex behavior collapsed in a cave in South Africa. And the passage Tucker squeezed through to find them is still so narrow that most researchers cannot physically enter it.
Number 12. Tardigrade. In 1948, Italian zoologist Tina Franceschi was examining a sample of dried moss scraped off a rooftop in Bologna. Under her microscope, she saw something move. This was strange because the moss had been completely desiccated, dried to the point of containing no detectable moisture for years. What she saw moving was a tardigrade, a microscopic animal roughly 0.5 millimeter long, shaped like a stubby, 8-legged bear that had just rehydrated itself back to life after years of being biologically dead. What followed was decades of systematic testing to find exactly where the limits of tardigrade survival actually were. Researchers found they survive temperatures of -272 degrees Celsius, 1 degree above absolute zero, the coldest temperature physically possible in the universe, to 150 degrees Celsius, hot enough to sterilize surgical equipment. They survive pressure 6 times greater than the deepest ocean trench. They survive radiation doses thousands of times above the lethal dose for a human. They survive the vacuum of open space. In 2007, the European Space Agency sent a batch of tardigrades into low Earth orbit on the outside of a spacecraft with zero protection. They came back alive. The mechanism is called cryptobiosis. The animal turns off every metabolic process, replaces the water in its cells with a sugar-based glass that holds the cellular architecture frozen in place, and waits. It can wait for decades. When water returns, it reassembles. The existence of tardigrades does not just challenge what we thought life could survive. It forces a genuine rethink of where in the universe life might currently exist, right now, in forms we are not looking for.
Only 1% of people know the dark truth behind what we are about to show you next. If you want to be part of the few who actually know what's happening in the world, subscribe and leave a like right now. Done? Look at this.
Number 11. Octopus vulgaris. In 2017, a team at the Marine Biological Laboratory in Woods Hole, Massachusetts, published a paper that the genetics community initially refused to accept as real. They had discovered that the common octopus edits its own RNA in real time in response to environmental changes. Here is why that is so disturbing. DNA is the fixed master blueprint of every living organism. RNA is the working copy the cell reads to build proteins, the machinery running every biological process. In virtually every organism on Earth, the RNA copy is faithful to the DNA original. Mutations happen in DNA and get passed to offspring. The individual organism does not rewrite its own genetic instructions during its lifetime. That is a foundational rule of molecular biology. Octopuses break it. When water temperature drops, Octopus vulgaris edits specific sites on its RNA, changing single molecular letters with surgical precision to reconfigure how its neurons fire. It is rewriting its own brain's operating instructions in real time, without touching its DNA, in response to its environment. The same individual animal. Within hours. The researchers found over 60,000 RNA editing sites in octopus neural tissue, compared to a few hundred in mammals. The octopus is editing itself at a scale with no parallel in any other animal on Earth. The implications go far beyond octopus biology. If an animal can functionally rewrite its neural code without altering its genome, then the relationship between genetics and behavior, one of the most foundational assumptions in all of biology, is far more complicated than science has ever modeled. The octopus did not read that model. It has been quietly ignoring it for 300 million years.
Number 10. Sperm whale language. For 30 million years, something has been sending structured messages through the dark beneath every ocean on Earth. Nobody was listening. Then, in 2024, MIT researcher Pratyusha Sharma published a paper that collapsed a century of assumptions about animal communication. An AI trained on 9,000 recordings found not the 21 simple click patterns scientists believed existed, but 156 distinct codas, each with 4 adjustable internal dimensions. That architecture has a specific name in linguistics: duality of patterning. For 60 years, it was considered the single feature that made human language categorically different from every other form of animal communication on Earth. The AI found it a mile underwater in an animal we had been hunting to commercial extinction. UC Berkeley linguist Gaspar Bacchus then slowed the recordings down and found that sperm whales produce vowels, sounds acoustically identical to human speech, using anatomy that evolved for echolocation, not language. The same organ producing those vowel-like sounds is the spermaceti organ, the system that also generates 230 decibels of sound, the loudest biological noise ever recorded, physically capable of vibrating a human body apart at close range. In 2023, off Alaska, a humpback whale named Twain held a 20-minute back-and-forth exchange with researchers on a boat, waiting for the correct conversational pause before replying every single time. And out of all 156 sperm whale codas, researchers have cautiously identified one they believe they understand: 2 clicks, a pause, 3 faster clicks, the most common coda across all Caribbean sperm whale families. Their best translation: I am here. It has been transmitted millions of times across 30 million years. We just built the machine to finally hear it.
Number 9. Chicxulub Crater. For most of the 20th century, the extinction of the dinosaurs was one of science's great unsolved mysteries. Dozens of theories competed. Then, in 1980, physicist Luis Alvarez and his son Walter published a paper the scientific community dismissed as absurd. They had found a thin layer of iridium, a metal rare on Earth but common in asteroids, in geological formations worldwide, consistently deposited at exactly the same moment in history, 66 million years ago. Their conclusion: a massive asteroid had ended the dinosaurs. The geological community rejected it for nearly a decade. Then, in 1991, the Chicxulub crater was confirmed, a 180-kilometer-wide impact structure buried beneath the Yucatan Peninsula, precisely dated to 66 million years ago. The reconstruction of what happened is the worst single day in the history of life on Earth. The impactor was 12 kilometers wide. It hit at 20 kilometers per second. Everything within 1,000 kilometers was vaporized instantly. Ejected material re-entered the atmosphere worldwide at hypersonic speeds, heating the air hot enough to ignite global wildfires simultaneously. The resulting debris blocked sunlight for years. 75% of all species on Earth were gone within a geological instant. But here is what genuinely disturbs researchers. The Chicxulub impactor did not just cause a mass extinction. The dust cloud that killed 3/4 of all life on Earth also blocked enough sunlight to collapse the food chains that kept large reptiles dominant and created the ecological opening that allowed small nocturnal mammals to expand, diversify, and eventually produce primates. The catastrophe that ended one world was the precise mechanism that created ours. You exist because of the worst day this planet ever had.
Number 8. Tarare. In 1772, a boy was born in France whose existence medical science still cannot explain. He was known by a single name, Tarare. From childhood, he experienced a hunger so consuming that it functioned less like appetite. French military surgeons who studied him during the Revolutionary Wars documented everything. He could consume a meal intended for 15 soldiers and be hungry again within hours. His body temperature was abnormally high. He sweated constantly, producing an odor so intense that he could be smelled from across a room. His pupils dilated to an extreme degree after eating. His jaw could open wide enough to swallow objects the size of an apple whole. Army physician Dr. Corville subjected him to controlled experiments. In one session, Tarare consumed a live cat. In another, a live eel without chewing. He was documented eating puppies, lizards, snakes, and at one point a meal of 30 pounds of raw bull lung and liver in a single sitting, and asked for more. Despite consuming quantities that should have killed him repeatedly, he maintained a near normal body weight. The calories were going somewhere. Nobody could determine where. He was eventually removed from hospital wards after infants in the maternity ward began disappearing. He died at 26. The autopsy found a stomach of abnormal size, a body full of pus, and no identifiable cause for any of his symptoms. No diagnosis in the entire history of medicine has ever fully accounted for what Tarare was. He remains the most medically inexplicable human being ever documented, and no case remotely like his has appeared in the medical literature since.
Number 7. Antikythera mechanism. In 1901, Greek sponge divers recovering artifacts from a 2,000-year-old Roman shipwreck off the island of Antikythera pulled up what appeared to be a corroded lump of bronze. It sat in a museum storage room for 2 years before anyone examined it closely. When archaeologist Valerio Stice looked at it under magnification in 1902, he saw gears, precisely cut, interlocking bronze gears with no business existing in the ancient world. The Antikythera mechanism is now recognized as the most sophisticated technological artifact ever recovered from antiquity. Using X-ray tomography, researchers determined it was a hand-cranked analog computer built around 100 BCE that calculated the positions of the sun and moon, predicted solar and lunar eclipses accurate to within hours, tracked the 4-year Olympic cycle, and modeled the irregular elliptical orbit of the moon using a pin-and-slot epicyclic gear system, a mechanical solution engineers did not independently rediscover until the 14th century CE. It contains at least 30 distinct bronze gears. The manufacturing precision required tolerances historians had assumed were impossible without modern machining. The Antikythera mechanism does not just prove ancient Greeks could build it, it proves that a level of mechanical engineering sophistication existed in 100 BCE that then completely disappeared from the historical record for over 1,000 years. Whatever tradition of knowledge produced it did not survive. We have no idea how many devices like it existed, were used, and were lost. We found this one because it sank in shallow water. Everything else went somewhere else.
Number 6.
Ophiocordyceps unilateralis. In the rainforests of Thailand, Brazil, and Ghana, carpenter ants occasionally begin behaving in a way entomologists describe as the closest thing in nature to a horror film. The ant leaves its colony at an abnormal time. It descends to a specific height above the ground, consistently between 25 and 30 centimeters, finds a leaf vein on the north side of a plant, and clamps its mandibles around it with a grip so powerful the muscles lock permanently. Then it dies. About a week later, a stalk erupts from the back of its head and releases spores directly downward onto the forest floor below, where other ants are walking. The organism responsible is Ophiocordyceps unilateralis, a parasitic fungus that infects the ant's body, hijacks its motor system, navigates it to the precise microclimate optimal for fungal reproduction, forces the biting behavior that anchors the body in place, and kills the host at the moment most useful to the fungus. Research by David Hughes at Penn State University revealed something even more disturbing. The fungus does not infect the ant's brain. Brain tissue in infected ants is found consistently intact. Instead, fungal cells infiltrate the muscle fibers throughout the body and control movement directly, bypassing the nervous system entirely. The ant's brain is alive throughout the entire process, a passenger in its own body, fully conscious, unable to override what the fungus is making it do. Hughes described it as the fungus using the ant as a vehicle while keeping the navigation system switched off. There are over 200 species of Ophiocordyceps, each specialized to a single host insect. They have been doing this for at least 48 million years. The fossil record contains amber-preserved leaves with the exact same bite marks.
Number 5. Denisova Cave. In 2008, a Russian archaeologist working in Denisova Cave in the Altai Mountains of Siberia found a fragment of bone, a single finger bone from a child, roughly the size of a large grape. It was sent to geneticists Svante Pabo's team at the Max Planck Institute in Leipzig. What they recovered from that fragment in 2010 was the complete genome of an entirely unknown species of human, one that had never appeared in the fossil record, had never been hypothesized, and had never been named. They called them Denisovans. Every previous hominin species had been identified from anatomy first. The Denisovans were identified from a genome before a single complete skeletal feature had been described. But the finding that genuinely disturbed researchers was what the genome contained. Between 3 and 5% of the DNA of modern people from Papua New Guinea, Australia, and Pacific Island populations is Denisovan. A species completely unknown to science until 2010 left a permanent genetic signature in hundreds of millions of living people alive today. A gene variant now found in Tibetan populations, EPAS1, which allows humans to survive at high altitude with critically low oxygen, was inherited directly from Denisovans. It is the reason certain Tibetan communities can live and work at altitudes that would kill most humans within days. A species we discovered from a child's fingertip in the Siberian cave is the reason those people are alive right now.
Number 4. Helicobacter pylori. Until the early 1980s, the entire medical establishment agreed on one undisputed fact. Stomach ulcers were caused by stress, spicy food, and too much stomach acid. It was a multi-billion-dollar industry of treatments and surgeries. Then, an unknown Australian doctor named Barry Marshall noticed a spiral-shaped bacterium in the stomach linings of ulcer patients. He proposed the unthinkable. Ulcers were not a psychological reaction to stress, they were an infectious disease. The medical community laughed him out of conferences. They insisted no bacteria could possibly survive the highly acidic environment of the human stomach. Denied funding and forbidden from experimenting on humans, Marshall did the only thing left. On July 17, 1984, he took a beaker containing billions of cultured Helicobacter pylori bacteria and he drank it. Within days, he developed severe gastritis, the agonizing precursor to an ulcer. He then quickly cured himself using standard antibiotics. In a single, desperate act of self-experimentation, Marshall proved that the entire medical textbook on gastroenterology was fundamentally wrong. His discovery did not just win him a Nobel Prize, it cured millions of people of a chronic condition that doctors had been blindly blaming on their lifestyle. It proved that sometimes the medical consensus is not based on evidence. It is just an assumption waiting for someone crazy enough to swallow the proof.
Number 3. Ötzi, the Iceman. On September 19, 1991, two German hikers named Helmut and Erika Simon were descending a mountain pass in the Alps when they spotted something protruding from melting ice. They assumed it was a discarded doll. Austrian authorities initially treated it as a routine recovery. Then, the carbon dating results came back. The body was 5,300 years old, the best preserved natural human mummy ever discovered, perfectly frozen since 3300 BCE. What 30 years of forensic analysis revealed about Ötzi's final hours reads like a criminal case file. He had eaten a large meal of red deer, ibex, einkorn wheat, and bracken fern approximately 30 minutes before his death. He had healed fractures on his right hand, consistent with a defensive wound, the kind sustained when someone grabs a blade. His stomach contained the DNA of at least 4 other individuals, none of them relatives. And lodged in his left shoulder was a flint arrowhead that had severed his subclavian artery. He bled to death from behind, shot by someone he likely knew. The oldest cold case in human history. But what stopped geneticists was his DNA. His Y chromosome haplogroup, the direct paternal line, belongs to a lineage found today almost exclusively on the island of Sardinia. A man who died in the Alps 5,300 years ago carries genetic ancestry that draws a direct line to a living population on a Mediterranean island right now. Ötzi did not just give us a window into Copper Age life. He gave us a thread connecting the deep human past to people who are alive today. And whoever shot him from behind and left him in that glacier never expected anyone to find out.
Number 2. Turritopsis dohrnii. In 1988, a marine biology student named Christian Sommer was collecting small organisms off the coast of Rapallo, Italy, when he noticed something strange about a tiny jellyfish he had placed in a petri dish. After reaching full maturity, the jellyfish did not die. It shrank. Its body reversed course, contracting back to its earliest juvenile form, a state called a polyp, and then began growing again from the start. Sommer brought his observation to researchers at the University of Genoa. It took nearly a decade of study before the scientific community accepted what was happening. Turritopsis dohrnii, a jellyfish measuring roughly 4 millimeters across, is the only known biologically immortal animal on Earth. When it ages, is injured, or is placed under environmental stress, it can reverse its entire developmental process, turning mature, specialized cells back into unspecialized ones and rebuilding itself from scratch, indefinitely. The process is called transdifferentiation, and it violates one of the core assumptions of developmental biology, that cellular differentiation is a one-way process. Once a cell becomes a muscle cell or a nerve cell, it stays that way. Turritopsis dohrnii does not follow that rule. A 2022 study from the University of Oviedo sequenced its genome and found specific gene expressions associated with DNA repair, protection against aging, and cellular reprogramming that operate at levels dramatically higher than in any other animal studied. The jellyfish is not just surviving, it is actively running anti-aging processes at a molecular level that science does not yet fully understand. Biological immortality is not a concept. It is a 4-millimeter animal currently living in every ocean on Earth, and it has been doing this for hundreds of millions of years while we were busy assuming it was impossible.
In January 1951, a 31-year-old woman named Henrietta Lacks walked into Johns Hopkins Hospital in Baltimore complaining of abnormal bleeding. She was diagnosed with an unusually aggressive form of cervical cancer. During her treatment, a surgeon named Howard Jones removed a sample of her tumor tissue without her knowledge or consent. That sample was handed to cell biologist George Gee. Every cell line researchers had previously attempted to grow outside the human body had died within days. Henrietta Lacks's cells did not die. They divided, and divided, and divided again, doubling every 20 to 24 hours, indefinitely. Gee named them HeLa cells.
They became the first immortal human cell line in history and have been in continuous use in laboratories worldwide for over 70 years. The Salk polio vaccine was developed on HeLa cells. The research that led to the HPV vaccine ran on HeLa cells. Foundational cancer biology, HIV research, the study of radiation effects on human tissue, all of it passed through HeLa cells at some point. There are currently an estimated 50 million metric tons of HeLa cells in existence, more biological material from one human being than from any other person who ever lived. Henrietta Lacks died on October 4, 1951, 8 months after her diagnosis. She never knew her cells had been taken. Her family was not told until the 1970s. They were never compensated. Her cells have been bought and sold by pharmaceutical companies for decades and generated billions of dollars in commercial and medical value. In 2023, a federal judge ruled that a biotechnology company had violated the rights of her estate. The woman whose cells may have saved more human lives than any other single person in history died not knowing any of it had happened. She never gave permission. She never received a dollar. And the science that runs on what was taken from her without consent is the science that is keeping people alive in hospitals right now, today, all over the world.
If you thought this was terrifying, you are not ready for what they found in the video on your screen. Click it right now. The truth is worse than you think.